Rock on. Oh, Mary, just making sure there's nothing in C, correct?
Correct.
Okay, yeah. I need to double check, sorry.
No, you're good.
Went off the assumption that it was open. All right, now we have something in C.
And that was 087.
Yes.
Roger.
Thanks, Kim. All right, now for brachiopod slurp time.
Yep.
Kim, would you turn on the porch light, please? Thank you. I really liking my grip here. Oh, all right, that gotta do. That'll do. Kind of. All right, I'm ready for the flush. We want to get visuals on it. That ain't it.
That was it.
Nice. Nice. Mary, what sample jar are we going to?
We can do 2.
2, roger that. Yeah, someone's home.
Huh.
Marley, want to push a note there, please?
Did that come up with when we tried to do
That's great.
that coral earlier, I think?
Oh, possibly.
Bycatch.
All right, we can go with 3 then.
3, Raj.
All the other.
On 3.
Roger that. And Tom, this is the correct sample you want here, this bivalve?
That is correct.
Okay.
That's the brachipod.
Brachiopod, sorry. Yeah. All right, Kim, go ahead.
Doesn't matter what you call it as long as you collect it.
Good, like that.
You can make up any name you want. The round white
right.
thing.
Should be in there now.
There it
There
is.
we go. Marley, full wide, please. Anything else you want slurped up here, over here?
Well, we want to pick up, if you look below the slurp right now, there's an orange
Seafen?
cup coral. It's a desmophyllum. So that could just be a grab, or it could be a slurp. I don't think it's attached because I think it's in the rubble.
Is it
I
the
don't
one
know.
below the slurp right now?
Yes, it's the orangey, reddish looking one.
Marley, go ahead and zoom on in there, please. Yeah. Yeah, we can go ahead and get this. I'll have to back up a little bit, I think. I don't think I'll get a very good angle on it. Actually, Marley, you want to come a little wide? A little bit farther wide, please. Maybe full wide.
And just to check, the brachiopod was sample 88.
Yes, it was.
Yeah, I don't think I'll be able to get that very well.
Thank you.
Yeah, I won't have the right range on that.
It won't bend enough.
So I put the bending strainer leaf on for a reason, I suppose, but all right. I'll back up a little bit and we can take that sample.
Okay, so before you back up,
Raj.
you can put the slurp away from it. We just want to look around because they want 4 to 5 of that kind of coral.
Okay.
So that one just happens to be alive, but there might be some dead skeletons. Don't really see any because they're fairly wide.
Not to interrupt, but a quick good night to my nephew and niece, Maya and Andrew.
Good night.
Well, good night, guys. Have dreams of Ophelia.
It looks so close, actually. I can just nodule it with the nozzle, sorry. Tom, I have a slight idea if we want to collect this without moving.
Okay.
I'm just gonna pick it up here and nudge it. We're pretty close to it anyway. And we want this in the same sample jar as the one before?
Could we put it in a different sample jar?
Roger.
And then we'll put all the Ophelia in its own sample jar. I mean, all the Desmophyllum in its own sample jar.
Sure thing.
Say when.
Go ahead. I think we got it. Oh. Okay, you want to kill the suction? Or reduce it? I think we're going to clog the hose. Marley, push in there, please. Yeah. I'll probably clog it. I can put this in one of the bio boxes here.
Sure.
Mary, is there anything in the toolbox?
In A, we have some urchins. so we can go with B.
Sure. Full weather, Marley. All right. Oh yeah, go ahead, please. Go on. All right.
Nice.
So we can just scan the rubble for other corals just like that, these desmophyllums.
Sure thing. Is there any that you see here in the view, or should we pick up and hop around? Tom, do you guys see any of the desmophyllums in this view, or should we hop around?
Are they looking for the
Sorry.
dead Ophelia?
Yeah, Tom, can you reiterate what type of coral?
Yeah,
like
sorry.
Yeah.
I got kicked off right at that time when you're asking me a question. So,
Oh, sorry.
yeah, so we're looking for, if you look on the wall, the living coral up on top, you see the two kind of orange ones, two orange fan ones. That's the type we're looking for. So they're going to be much thicker, much larger structure than the Lophelia with its thin type of structure. So we're just looking for something bigger, fatter in the rubble. I don't want to pull them off the wall if we don't have to.
Roger. So the ones that
And
are...
I haven't,
Sorry.
I didn't see any right here, right now. I saw that one live one, but I haven't seen any others that are really popping out at me.
Okay.
So we can look at another place. We did see some farther down the rubble when we were coming up, so.
Sure thing. So we can go downstream.
Yes, we can.
Roger that.
Are you backing down the river?
Backing down the river. So you said there's some down here that you had seen before?
I'd seen them before, yeah. Now finding out exactly where I saw them is the next question.
All right, we'll use your eyes.
So maybe if we just stop and zoom in
Sure,
for a moment, maybe we'll be able to see it better.
sure thing. We can park ourselves somewhere and
There's
probably
one
park right
bottom
here.
screen right now. You see?
Yeah,
Oh,
I think
yeah.
so.
You guys want them live or dead, though? I think is what they want dead.
Just not attached to the rock. I was trying to get them that had fallen off.
I see.
Of course, trying to find the same one that you found on the way up doesn't work.
Is this one here kind of by the lasers? Is that one
If you zoom in, I think it might just be a regular cup coral and not a desmophyllum.
Oh, hey,
They're
Marley.
more oval.
Marley, want to... Yeah, thank you.
Yeah, that one's not oval.
Roger.
It's round.
Roger that. Go ahead and come wide, Marley.
If it comes to it, we can pull them off the wall, but I was just trying to be good. Find a skeleton instead. Can we look to the upper right? If we can zoom in, I think I see one up there in that rubble.
Sure thing.
You're so accommodating.
I'm just driving a lens here. You guys are doing the ones that are doing the science. Marley, go ahead and zoom in here on this rock.
And if we could pan, yeah.
One of these here?
Can we pan up a little more?
Sure. Little
So
more?
right there between the lasers,
Oh, the dead one there, yeah.
the dead big guy, yeah.
Okay.
Tom, did you want
You
to
gonna
count
come full wide there,
this as
Marley?
one sample or two samples?
Lizzie says 4 to 5 samples.
Okay.
So we have to find a few of them.
I just meant, did you want different sample numbers or is one sample number okay?
Oh no, I'm sorry, this will be one sample number,
Okay.
yes. And we'll just put the number of pieces that are in there.
Just wanted to make sure.
And you can just grab the rubble and throw it in.
Sure thing. You want it with the the one that we just kind of
Yeah,
pseudo
we'll put them
slurped?
all in the same bio box. Yep.
Roger. Tom, you have eyes like a cat. This is great.
Sometimes. From watching hours and hours of video.
All right, Ben. We'll see how we settle. On the little marble, yeah. All right, Ben, one more time.
Looks better.
All right. You want to zoom on this guy before we take it, or you just want to take it?
I think we can just take it.
Roger that.
If you're going to be accommodating, I'm going to be accommodating.
Turn down the grip force to non-squish.
Was that an official term, non-squish force?
It is indeed an official term. Brought to you by the 4 to 8.
The fun watch. That little fish wants whatever you do.
Yeah, he remembers that sample from when he recruited to it, Tom.
Yep. You can see on the
I can-
Desmo how it has all those plates inside of it, and it's oval shaped. That's how we can find them. You can see the other one looks the same.
Reg.
Nice job.
Yay.
Nicely done.
Now we got to scour, look around, see if there's two more.
I'll need your cat eyes.
Yeah, well, I see something bigger to the right of the fish.
Right of the fish, Reg.
Somewhere in there. Yeah, just about to the right of the right laser.
Go ahead, Marley. Push on in, please.
Get right under there.
Oh, wow. That looks like one.
I think it is. I think it's just upside down
Yeah.
because it's growing way thicker than the other things.
Those are some really good eyes for catching that.
I was just about to say that.
Would you like this one, Tom?
Yep, let's try that one.
Roger that. Go ahead and come by, Marley.
I'm basically just looking for things that look different in the bottom.
Did you see two there or just one?
There's two.
I
Like a
just,
bigger and smaller one
Yeah.
together.
oh, I see now. I only saw one. My eyes only can see one at a time. So.
Together we make a very, very good set of cat eyes.
That's good. At least we don't have to do it in the dark.
Marley, you want to push in there, please? That's great.
Oh yeah, I see the second one now. Yep.
It's pretty small.
Yeah.
Yeah. Let's think about this for a sec.
Mm.
It looks kind of live.
Would it help to grab extra of the Ophelia Rebel?
Yeah, you can grab some extra Lophelia and
Roger.
it starts running away.
Pull either, Marley. Let's check to see if we have it in this handful. All right, go ahead, Marley. I think we still have it.
Yeah.
Yeah.
I think it's there. It's right next to that one finger.
All right, full wide. No, no worries. All good.
So when you set down like this and you use the manipulator, do you put Herc in auto? Is that how he stays down?
It's something called stick lock.
Okay.
Basically it means that we'll, here we go. Big subsea scoop.
Nice job.
So I think there was another one after you pulled that up. I think I saw one in the rubble. Like we exposed one. So if the fish didn't move, we'll know where to look. But
All right. That fish is a good marker.
Fish.
so just try to zoom in there and
Go ahead, Marley.
see.
Want us to look for somewhere else there, Tom? Oh, did he drop off the link again?
Nope, I had it on mute. So that's the other problem. Yeah, they were just picking up the garbage, so you didn't want to hear the garbage truck go by.
Oh, Raj.
So the kind we're looking for, if you look up on the rock wall in the upper left corner, you see that pinkish one.
If you look up a little
Now
more.
it's dead center. Actually, there's a dead one right there.
Oh yeah, big.
Dead center, that one?
Yeah, we could pull that one.
Sure. All right, Marley, one come pull wide. All right, Marley, want to push in a bit, please? That's great.
Just knock her off.
I don't know if this is going to work out the way I want it to, but we'll try.
Scrape it.
Hopefully I got it still. Yeah, buddy.
Oh, nice. Little hammock.
Just cradling it.
All right, that was cool. All right. Pull wide, Marley. All right, Kim, I'm ready for you.
I believe that's 4. So she
Yeah,
said
Kim.
4 to 5, so
Yeah, thank you.
I think we'll be good with 4.
Roger that. The cheer test will be actually getting us in the box.
Yeah, Lizzie's off. She's lost internet connectivity right now, but I think 4 is perfect.
That's what I want to hear. Oh, he's going to lose his hammock.
It's okay, he'll find a new home in a petri dish.
Nice.
Oh,
Don't think I've ever seen that before, Jeff.
I haven't done that before. It's fun.
All right, I think we're good. We might actually be able to leave the spot. And Meredith's saying how she loves how the urchins run around the bio box.
So, our plan for leaving the spot, do you want to keep along this, like, bouldery feature for a little? Kind of go...
Yeah, but the kind of the way you were showing me before, along that axis, and then
Yeah, so if you've seen high pack...
basically,
yeah.
we're gonna, yeah, I've got, right, go ahead. I've got high pack open.
Yeah, in high pack I have a little green arrow. I'm thinking I'll get us off this boulder towards, it's going to be 320 is going to be the bearing from here, if you see the little green. And it may be a little downslope, but just can kind of explore. It's only 150 meters. And this will cover from the middle to the northern part of this. And then,
okay.
And then you want us to skip C, D and just go straight to C, E? Is that?
Yeah,
Yeah.
and that's correct. And could you zoom out so I can see? Yeah, there we go. You see that feature over just south of CE with the that kind of bump.
Oh, this very small contour?
Yeah.
Yeah.
So I'd like to go kind of straight up over that to
All
CE.
right. So that sounds good.
And somewhere in there, we're going to want to run another transect. Is it? Could you give me a ballpark of the distances?
Yeah. So if you look that temporary target below that little mound, that's 235 meters about. So we have just enough to transect kind of. Yeah, I think this, this is a good bearing to hit everything. We just might go a little downslope, so just might keep a certain heading as we come down that mound and then maybe go straight forward, if that sounds reasonable.
Yeah. Tom, does that sound all right to you?
Yeah, that works because we never know what we're going to encounter. So if we're already foreseeing that could be a problem, that's way better than ending up on a ledge that we never saw.
Right.
Roger that.
All right, so just make sure, Jess, I'm thinking 320. Just, yeah, I think 320 is good.
Roger, 320. I'll get in front of Argus and then we can call in those moves.
Roger.
UTC.
You don't have any wraps from all the pure wedding?
No, although I'll probably put one in right about now if I keep turning that way. Let's just go the other way.
It's amazing. And none of the other rocks have the Lophelion. Just the one at the top.
Definite zonation here. So do the rockfish associate with the corals do they forage on them, or it's just?
The rock, I don't know if they would actually eat a coral, but there's definitely a lot of things like coprolids and amphipods that live on the coral that they would eat.
So all the associates are good for them.
Yeah.
Cool.
But who knows? Go ahead, Chris.
So that's 320.
Yeah, that's 320. And I'll just hop up here and get myself set up for the transect. All right, I'm ready for
Look
you
at the
there.
pretty flag rockfish.
The striped thing? Super
The
striped.
red and white one is the flag rockfish.
This is transect 4.
That sounds right.
Okay, you ready?
Right, I'm ready. Let's do it.
Are lasers on? Or am I just not seeing it?
Oh, no, Mary, but you
Mary.
are so on it. It's great. Thank you.
That's helping those cat eyes, see? Yeah, there are the cat eyes right there.
I think it's interesting there's so many species of rockfish in the same spot. Like, there's no niche differentiation, or at least spatially, you know? Right? Like, they're all different species, but they're all aggregating in the same spot with similar morphology. And,
Yep.
Yeah.
And all sorts of different colors, and a bunch of them feed differently. So like the widows will feed up in the water column more. Some probably specialize on other organisms. You know, like one might like crustaceans, one might like worms or whatever. But most of them, I'm pretty sure, are opportunistic. So it is interesting that you have all these different ones together. What caused them to speciate into all these different colors and morphs and just everything that's weird about them.
Starting the
You see the flag. Thank you. You see the flag with his vertical stripes, and then you see other ones like a green stripe with horizontal stripes. And this brown one in front of it is a square spot.
It's like diversity for the sake of diversity.
That's what it seems like sometimes.
I guess they're successful.
Do they all have different feeding habits? Are they all like feeding on the same things mostly? I know it's
some are
opportunistic.
different, but yeah, so some are different, but a lot of them eat the same things. So some of it might be size related, that they're eating smaller things or larger things. Some of the bigger ones are eating the smaller species.
Looks like we're just a little bit to the eastern side of the strike of this feature. But
Roger that.
So that's a speckled rockfish there, that brown one. Looks like a little coral to the left. Could have been Caligorgia. A few sponges. Poecilastra, or however you say it, I just call it Bo.
Get on top of this feature a little bit.
So I was mentioning earlier when we surveyed this area with sonar, we could see the seafloor and how complex and rugose it is, what we're seeing now on the video screen. But we also had a sonar looking for fishes, and we saw that there were just a ton of fishes. So this is a marine reserve, a marine protected area off of the Channel Islands in Southern California. And our sonar would detect all these fishes in the water column, but we couldn't see what they were. We didn't know what species and how many of them there were. And so the only way to do that is to get under the water and put eyes on it, which is easy to do in the shallow water environment where you can jump in with your scuba gear and survey. We've done that a bunch, but that gets you down to about 30 meters. And about 90% of the Channel Islands National Marine Sanctuary is deeper than 30 meters. It goes down to nearly 1,800 meters south of the islands in the Santa Cruz Canyon. And so to study and understand what's going on in the other 90%, we need other technology. So we have a great partnership with the Ocean Exploration Trust and the Nautilus team here to survey and understand what's going on in that other 90%. So where are the fishes, where are the habitats? And if we can understand both those things, where are the corals, the sponges, we can better manage the resources to ensure that we can sustain them for future generations. So that's a good part of what all these studies are doing. We're collecting samples, as you may have seen just earlier, brachiopods and corals. We'll take those back and working with partners at different places on the East Coast and the West Coast locally. We're working with the Santa Barbara Museum of Natural History. We will take those organisms back and identify them in a lab and make sure we've classified them and understand what species we've captured in our sanctuary. And we can monitor them then for change, understand what's here and what's changing as our marine ecosystem changes with climate, from changes in sea level rise and sea surface temperature and ocean acidification. What are the impacts, and also different pressures? This area here is near Richardson Rock, which is a very diverse and fish prevalent area. Great, all these rocky, rubbly features that you see here form fish habitat. Areas where the little juvenile rockfish will recruit and can find shelter from larger species that might eat them under all those nooks and crannies that you're seeing. And the older individuals will also spend time there looking for food as well. And so right now we're collecting data along a transect. We're moving about 200 meters distant, and we'll count, enumerate all the deep sea corals, the sponges, the fishes, get a better idea of their densities and how those populations are doing. That'll allow us not just to understand fish and fisheries, which are important for the Channel Islands, but also just biodiversity and what's going on with our local marine environment.
And here's more Lophelia coming up.
Some pretty neat terrain.
It is. These are some big rocks just hanging out here. Yeah. And one of the interesting things about rockfish is many of them go to sleep at night. Oh. So you can start looking in the holes, and you'll see a lot of rockfish hiding in the holes as they are finding a place. As it gets darker, they're finding a place to hang out for the night. So we won't see as many rockfish as we go along. They'll be sitting wedged into the bottom.
So only the mesopelagic species do that? Or do the deeper ones also have cycles like that?
Most of the rockfishes go through that, even the deeper ones. But there's a few species that actually do the opposite and come out at night or come out more. A lot of them feed crepuscularly, so they do it at dawn and dusk. And the rest, that's why we see them just hanging around in the middle of the day, doing nothing.
So Tom, when we surveyed this area, when we multi-beamed this area to collect the bathymetry, we were just going back and forth as you do in the boat to collect your bathymetry data, and we were doing it for a week or 2 weeks. And we just back and forth, days on end, nights on end, and we simultaneously had the fish sonar EK60 running to look for where the fish were. And all the surveys of the seafloor that we did during the day, we saw loads and loads of fish. And the ones we did at night, of course, we didn't because they were so close to the bottom. The sonar wasn't picking them up, just like you're saying. They were going to sleep. And so it looks like we have these horizontal east-west bands of fish populations because we only saw them during the day and not the evening.
Super cool data.
Yeah, that's what
super relevant.
When we did Express, that's why Liz wanted to do like one vehicle all day and the other vehicle all night. So what Express is we did 2 cruises along the coast over the last 2 years for 30 days. We had an AUV and an ROV on board, and we had to separate them. So the ROV basically went during the dark hours and the AUV during the light hours, because if we would go over the light-dark change, then our counts of fishes would change. So if we did nighttime counts, that's fine. We're just looking at ones that would be out at night. But if we're doing daytime counts and then we go overnight, then we lose. The counts during the dark would be completely different than the ones during the light. So the things that you have to consider when you're planning cruises.
It's an interesting way to do it.
But when you're looking for corals and sponges, they don't really move, so you don't have to worry.
Unless Jess comes by and grabs them.
That's right. They get to live in their little hammock.
So you prefer to separate out like the operations to each vehicle day and night versus like subtracting out an average or something?
Well, if we were doing something like here where we're going to go over day and night, we would only use the fish counts for like assessments for the daylight hours. Because it's going to be different at night. Yeah, there's a line coming.
Yeah, one's going down. All right.
Oh yeah,
that's pretty good.
Lizzie's pointing out that that's the 3rd or the 4th line in this MPA. But those lines look old, so they may have been here well before this was an MPA.
They better be.
Well, there are people who do go into these, so.
Oh yeah.
Hopefully they all get caught, but there are fishermen who will. They'll skirt the line and then maybe go inside just a little. But luckily most of the fishermen obey the rules because otherwise there won't be any fish left for the next generation. So the fish down at the bottom, you can see them hiding in the rock already.
Sorry, Tom, I'm just gonna make sure we're clear of all these lines here and then continue. Looks like this is right on our transect.
No worries.
That looks fine.
Looks like we have some Placagorgia coming up right where the vermilions are on that rock in the middle.
All right. I don't know if you guys wanted to mark that as a pause in the transect. All right, Kim, you want to come
We'll
back
just
to
do it after.
Kim, you want to come back to 320, please?
You just came like 15 meters off to the north of it.
Roger.
Now we're back on the mud with the green striped rockfish.
Yeah, we just get off this feature, it looks like in the map too, so it's good. We should end this right before that little mound.
That's fine. One of the things we do is when we post-process this, we look at the habitat data. So we will actually subdivide the corals and sponges and fishes by habitat. So we still need mud too, even though a lot of the things we're targeting are corals and sponges on the rocks.
Yeah, I guess it would be pretty biased if you're only looking at the rocks.
Yep.
Only
That's one of the
squid,
neat
please.
things. What was that? Excuse me?
Only giant squid, please.
Yeah. If we find that giant squid, we can just stop the cruise right there.
Right.
That's it.
1,000 zooms, please.
The day they find a giant squid, you're not going to be on board. The way you talk about a giant squid.
Isn't that usually the case?
Yeah.
Oh, I just left the room and all of a sudden we came across X and it was so great.
That happened with the whale shark to me in Hawaii.
Yep, that would be.
That line's off to this.
Yeah, I
off
see.
to the left again.
Its bearing is 320.
Yeah, that line looks like it's been here a while. It's covered under the mud. As long as it doesn't come out.
That is one of the things we try and note here, is marine debris where we see it. I think it was last year we came across a kilometer or so of net draped over our reef on the south shore of the marine sanctuary, and it can take a long time to break down and have an impact on the ecosystem, the ability of corals and other things to grow, and so we try and track that down and figure out where we have issues with things like marine debris and how to clean it up. That's everything from nets and lines, derelict traps, or plastic stuff that folks that comes in from the land, makes its way out to sea and can settle in some of these habitats and affect the fishes we eat and the biodiversity that surrounds the islands. Looks like we've got a little reef coming up here in our front.
So is that the basket star, that giant brittle starry thing?
Yeah, that's the basket star.
Huge.
Definitely. All its tentacles are out collecting all the plankton.
Do you have another ship moving, or?
Oh.
Too busy talking about giants.
Mostly. How far are we?
Gorge here to the right.
Yeah. So we have 50 meters left. So it'll be pretty immediate.
Nice size vermilion rockfish.
You are correct.
I did my early work in the Caribbean, and I think I knew every species of fish there by name, but it's a whole different ball of wax out here, and it's great to be working with you, Tom.
Well, if you really want to learn them, you can watch video for a while.
I am, and I keep looking up every species that you mentioned.
And we have a sculpin to the bottom right, sitting out on the mud. He's just at the bottom of my screen. Now he just went off the bottom of the screen. There he is. Looks like a rough back sculpin. Using my cat eyes, I can see the long spine on the front of his torso. It's white. You can actually see it. It's a white line coming up from the back of his head.
They're up to speed now.
Sorry?
They're up to 0.3 knots again.
Roger.
And here's some more black gorgia coming up. There's coral on the right. And we have a pink surf perch. It's the deepest living surf perch out of all the ones because there's no surf around here. Yep, they live out on the mud, so this is the place to find them. Most of the other ones that you are up in the kelp bed or right in the surf. The other species, not this species.